Cool. Okay. So yeah, thanks everybody. Good evening to everybody in the UK and good morning from New Zealand, or should I say kia ora. So I'm going to talk for about 15 minutes now about a project that we did last year, sadly before before Kerry's talk research that she just talked about was added. That would have been amazing to have that in this as well, where we interrogated the National Earthworm Recording Data Scheme datasets to look at earthworm distribution and habitat associations. So I won't go into this. I can skip a few slides because there's been lots of talk about the recording scheme and the lack of data. So you've seen this already in Kieran's talk, but just to say earthworms are poorly recorded historically. And we set up the national, I didn't, but the National Earthworm Recording Scheme was set up to fill in some of those gaps. And there's already been some talks today about the types of records that we have, so I can breeze through this one as well. So, but just to say that this project looked at two types of earthworm record in the National Earthworm Recording Scheme. There was the public records, so in this we'll call them earthworm recorders, and that's if you come on one of our courses or you submit some records, then that would be added to the to one part of the National Earthworm Recording Scheme and the public records. And then there's also these research records, which we've talked about today as well. Myself and others have historically got those from either approaching earthworm researchers in the UK or by transcribing records from the literature, as Kerry was just talking about. So we've got these two sets of data, and now we've got quite a lot of data that we can start to interrogate. So as of last year, so this is before Kerry's work was added to this, unfortunately. I wish we could go back and do it again now, but sadly, we all have lives and time must move on. But we had, at the point of this study, there was 21,500 records in the database. I think Kerry said there's now 25,500, which is amazing. And that's versus about 6,000 in the last publication that did this kind of work. So we've done a lot of work in that time, in about 9 years. There's a huge amount more records. And that's from 3,500 sites now versus 333 that were in the last publication. So we've had a lot of data. If we break down and interrogate it just really simply, we can see about a quarter of the data, the records are from the public, and about 3/4 are from research. So that's here. And we can look at national distributions and spreads, but Kieran's kind of already talked about that, where we're perhaps missing some patches, and some of those have been filled out. And I'm delighted to hear the Isle of Man is now represented in the data set, so that's great. And there's still lots of areas where we could do with more public recording and more research records, but that's for another time. What we did for this research that I'm talking about is three things. We interrogated the data set to look at patterns and biases between the public and research records to see if members of the public and earthworm research scientists are actually looking in the same places and using the same methods, or are we all doing different things? We could also start to ask questions. Now we've got lots of data, we can start to ask questions about habitat and microhabitat associations of earthworm species in the UK. And from all of that, we can hopefully try to start picking apart where some of the supposedly rare earthworm species can be found, and can we better target future sampling and research to learn more about the ecology and the distribution of earthworm species that we don't know a huge amount about. So I'll come back to this at the end and answer whether we were able to do this. Just to dive in, I won't go into the stats methods that we used because frankly, some of them are over my head. We had a statistician, colleague of mine helping with this, but we can talk about that if anyone wants to. But just to say, we interrogated those data, and now I'll present it over the next few slides, some of the most salient points. So here we've got, to start off with, a bar chart. On the left is the results from our study in 2002, and I'm comparing it here against the data from the previous publication in 2014 that did something like this using earthworm data in the UK. And I've tried to format that to be kind of comparable. And we can see now with the update, the addition of sort of, I don't know, sort of 16,000 records, more records, we can find a lot more useful information from the national distribution and proportion of earthworms in our data set. A few points to note is that firstly, we've added some new species. Quite a few new species have been added since 2014. There are some at the bottom here that were considered extinct or very rare within that data set that are now perhaps not so. So for example, Apoxodea limicola here was not featured in the previous national data set. It's now about 3% of the national records. So quite a big difference there. We can see that actually the top three most abundant earthworm species haven't really changed that much, but their relative proportion within the data set has changed. So we've learned a lot more about them, and probably in the top 10, things have swapped around a little bit, but certainly if we go to the top 15 or so, there's been quite a lot of change in how we view the commonality of some of these species. And that's important because some identification guides have been produced looking at the commonality and trying to find the most, based on the most common species you might find. And actually, with this updated information, it kind of blows a bit of that out of the water. So some things that we thought were previously common may not be, and some things, and vice versa. It also asks some questions about, now we've got a lot more data, we can say that some of these species which were previously considered rare or extinct have started popping up. And is it a case of they are rare, or are they actually just undersampled? And that's something that I was keen to try and interrogate further, and we'll come back to that a bit later in this talk. To pick apart the two data sets a little bit separately here and try and look for differences between the two. On the left here, where it says recorder, these are the public records, and on the right here, research is obviously researcher records. And this is interrogating them based on the method used and the type of habitat that those studies were sampled at. And basically, this is just a heat map which shows the number of sites within the data set at which these combinations featured. And one of the key things that immediately jumps out, I hope, is methodologically, there are some big differences between the public and the research and researchers. So for some reason, the public are not keen on, public recorders or amateur naturalist recorders are not keen on combining soil pit and vermifuge sampling, although that is a very common technique used by researchers. Quite a lot of sites and studies were using that. And you might expect to see different earthworms returned by not using that method. Conversely, researchers aren't particularly keen on microhabitat sampling. So deadwood and non-deadwood turnover, as well as compost and dung, are things that earthworm researchers are not prepared to get their hands dirty with, potentially. And so obviously we're going to have some major differences in the kind of species. If there are habitat associations with those, earthworm researchers are probably not going to be finding those as much as the public, and we'll come back to that. If we look instead at habitat type, so looking upwards on the columns rather than across on the rows, we can see that in both cases, heathland/moorland and scrubland are pretty poorly represented in the data sets by both research record types, but also wetlands are not very well visited actually by either group as well, to be fair. But here definitely in research, wetlands aren't used. So there's some major habitat types which are not occurring very much within the data sets. And if there's any earthworms that could be associated with those, we might expect that they would be poorly represented. And it's worth just pointing out that by far and away, farmlands and woodlands were the most commonly sampled sites in both groups. And particularly the combination of soil pit sampling in farmland made up a huge amount of research records. And that's because that's where all the money is, right? So if we move along, and apologies if you've got a bar at the top, I don't think you do, that blocks this off, but we can start to interrogate the earthworm species proportion within the data set based on things such as sampling protocol. And the arrangement of the species here is in this list corresponds to, if I just go back, it's exactly the same as this. So it is the most commonly occurring down to the least commonly occurring. So up here, we've got the most commonly occurring top 10 species were frequently recovered by soil pit sampling. Now that you might say that's because soil pit sampling is the most commonly used methodology, and you might be right with that. But just to say, quite a lot of the species were recovered by soil pit sampling, but certainly not all of them. And we do see some clear habitat specificity, or at least sampling specificity for some of these species. So for example, looking in deadwood, I mean, technically it's microhabitat. It's also a method looking in microhabitat sampling. And we see that species such as Dendrobina hortensis, Pygmaea, Dendrobina tempsi, Bimastus iceni in a huge way, and also Bimastus rubidus, were all quite strongly associated with deadwood sampling. We also find some clear compost and dung associated species, for example, so Dendrobina bonita, Icenia, Andre, and fetida, and Andre is definitely here, so that's great, are all associated with compost or dung. And you find some other patterns here. I haven't got time to go through all of these, but we do see some distinct methods and species returns. If we then interrogate these data by habitat, you see similar patterns emerging. So again, a lot of the most common species that we find are commonly returned by farmland. And I think, in fact, about 18 out of the most 20 common species in the UK were returned by both farmland and woodland the most. So we are catching, in farmland and woodland sampling, we are catching quite a lot of the species in the UK, but certainly not all of them. We do see some habitat associations that are very distinct. So for example, Heliodrillus oculatus, 80% of the records from that came from wetlands. And also this 20% here came from, this was wet woodlands, so riparian woodlands, I believe. And obviously Kerry's got some more of those records now. It'd be interesting to know where they came from as well. But we know that wetland sampling isn't taking place in any great degree. So we might expect that one of the reasons we don't see Heliodrillus oculatus very much is because we're not looking in the places where it is. We can also see some other associations with wetlands. So Dendrobina pygmaea came across in there, some Friendy, which is another rare species. Icenia tetrahedra and Octolacine lacteum were recovered quite a lot from wet soil. So we also see urban associations, and these will be corresponding largely to the composting species, I believe, but not exclusively. So we do see quite a lot of clear habitat associations. Then the question becomes, and please, nobody ask me any questions about the stats on this one. The question becomes, are the rarest species that we've got in the data set, or that we find nationally, are they associated with the most uncommon combinations of habitats and sampling protocols? So our statistician dug into this, and essentially this plot here, each point is a species within the UK, within the data set. And the further to the top and the right of this plot is the most common, the more common species and the more common habitat sampling protocol combinations. Conversely, towards the bottom left, you've got the least commonly occurring species plotted against the rarest combination of habitat and sampling protocols within the data set. What we find is a very, very strong relationship between this. And here we've got I think it's about 12 species, the most 12 common species occurring in the most common sampling method and habitat combination. And this would be farmland with soil pit sampling. And this would be our top 12 species in our list there in the previous graphs. Down here, we've got our 5 least common earthworm species, and they were significantly associated with the least common combinations of sampling. So here you might find things like one of these points would be Heliodrillus oculatus, and I'm pretty sure this would be wetland sampling, for example, and there would be others. So this suggests that, in fact, some of those rare species, or all of those rare species, are probably not coming up very much because we're not looking in the right places. And this is real food for thought for future sampling. So we will interrogate these data more, and we will come up with a plan for how we could best target future sampling methods to try and recover more information about those species ecologies. So just to conclude and try and answer some of the questions that I posed at the start, we did find that researchers and the public are definitely looking in different habitats, and they are certainly using different methodologies. We saw that most of the non-soil-based earthworm sampling was actually being carried out by the public. So we don't get a lot of microhabitat sampling from our research records by comparison. So keep it up. Everybody here who is looking in microhabitats for earthworms, please do it more. It's the only way we're going to learn about the ecologies of these species. We found that a number of species do demonstrate habitat specificity and also methodology specificity in terms of recovering them. And we found evidence that the in inverted commas rare earthworm species were associated with some of the least common habitats and sampling methods. So yeah, as I said, we're gonna look at this a bit more, and we're going to think about how we can best target future work to learn about some of these rare species and fill out some of the gaps on the map. Just to start wrapping up, because I think that's about 15 minutes almost. As it was mentioned earlier, this research I presented at the International Symposium of Earthworm Ecology in Rennes last year. And the manuscript was submitted a few months ago to the special issue for that conference of the European Journal of Soil Biology. It's currently under review. There's been some delays, I think, for quite a few submissions there, because it was a big conference with lots of people submitting papers, I'm sure. You'll see that here, the majority of the authors and co-authors are actually members of the Earthworm Society Committee. So we're all very keen to get that back ASAP and share it with you, and we can all use the lessons from it to start moving forward and learn more about earthworm ecology in the UK. So with that, I'll wrap up, say thank you very much for your time, and huge thank you to everybody who submitted records, whether it's research or the public. This is by no means an exhaustive list, but it's everyone I could fit on the slide. Yeah, if you see your name there, or even if you don't, but you submitted records, thank you so much, because it's enabling us to answer these really fundamental questions now about earthworm ecology. With that, I will be happy to take any questions.